Hello, welcome back to MLSQR Medical Lab Science Quick Review. And today our topic will be antibodies, structure, and function. And we'll have a question and answer at the end. Every day we are under constant attack by millions of mutating microbes that try to get through our innate and adaptive immune system. Appropriate B cell activation and clonal selection by a sophisticated mix and match genetic recombination of genes mobilizes a specific and targeted counterattack to keep these microbes at bay. Let's look at the five classes of antibodies produced by B cells that originate in our bone marrow to mature to plasma cells that become antibody factories of our body. The heavy chains are the two longer chains shown in the middle. The constant region of the heavy chains determine the biological function of each antibody class. Also note that the hinge region of the heavy chains between CH1 and CH2. CH stands for constant heavy domain. Two light chains are covalently connected to two heavy chains by two disulfide bonds. So the heavy chains determine the antibody class and biological function. Known as antibody isotypes, the immunoglobulins differ in the constant regions of their respective CH1, CH2, and CH3 domains. A memory mnemonic is gamed. The order of antibody class by serum concentration is GAMD. The first two, IgG and IgA, have subtypes. IgG crosses the placenta with IgG1, the subtype that is best at this function. IgG2 is the only IgG that does not cross the placenta. The good, passive immunity from the mother to the baby. The bad, hemolytic disease of the newborn when the maternal IgG antibodies attack the fetal red cells with the antigen. Details we shall see in a later video. IgG can cross the blood-brain barrier, so it's found in CSF. IgG is very effective in binding to specific foreign antigens in invading viruses and bacteria and neutralizing bacterial toxins. When IgG binds to its corresponding cognate antigen, it forms an immune complex, which can fix complement. Activated complement proteins form a complement cascade that forms a membrane attack unit that effects hemolysis. In the blood bank, we see that Rh antibodies, five of them, have a thermal range observed with red blood cell agglutination best at 37 degrees, but also detected at indirect Coombs phase with anti-human globulin or anti-IgG. Anti-Kel, Duffy Kid antibodies also react at the AHG phase, as well as anti-A, B. Unfortunately, IgG can also cause autoimmune disease. Multiple myeloma is a monoclonal gammopathy with neoplastic plasma cells producing too much IgG. In IgA, we see that there are two subtypes, 1 and 2. A connector called the J chain connects the two monomers, forming a dimer. And this is the passport that facilitates the transport of the IgA through the mucosa and to the intestine. And it resists acid digestion and enzymes in the digestive tract. Not only is IgA found in human serum, but it is the predominant antibody in body secretions, nasal mucus, saliva, tears, breast milk, and it is the most prevalent overall because of the secretions, forming a sound defense against foreign invaders in our mucus, in our nasal passages, and as well as in our gut. The mother gives passive immunity by IgA going through breast milk to the baby to help the baby fight infections. Giving plasma to a person with IgA deficiency can result in a severe anaphylactic reaction. IgM is a huge molecule because it's a pentamer. Like IgA, there is a J chain connecting the IgM pentamer together. When IgM binds to its cognate antigen, forming an immune complex, it fixes or activates the complement protein cascade. And it is best at this function because it has 10 binding sites, 10 active sites that will activate the complement and affect the membrane attack unit to affect hemolysis. IgM is the first to appear in a fetus and neonate, so the baby makes IgM. But note that the mother does not give IgM through the placenta to the baby. IgM is prominent in the early immune response, indicating early infection, as opposed to IgG, which appears later and stronger with a higher titer, with a fourfold rise in titer when you compare an acute and convalescent titer. In the subsequent infection, the B cells and plasma cells mount an even greater and quicker response, known as the anamnestic response, which you also see in the booster shot in vaccinations. IgM and blood bank, we note that the thermal range of IgM antibodies, they agglutinate red blood cells best after immediate spin at room temperature. IgM antibodies are enhanced after 15 minutes incubation at 4 degrees centigrade incubation in the refrigerator. IgM binds to the B lymphocytes membrane because of the hydrophobic residues in its constant region. Also, IgD will bind to membranes of the B lymphocytes. The ACP study guide states that the main function of IgD is in B cell development because it is the isotype of surface immunoglobulin of immature B cells. IgE attacks parasites and summons the mast cells with IgE receptors to release histamine and other chemicals in their granules to kill the parasite. Eosinophils are involved in eosinophilia seen in chronic parasitic infections, also neoplasms, allergies, and asthma. After IgE binds to the corresponding antigen on the parasite, the mast cell has IgE-Fc receptors, which will recognize the IgE bound to the parasite. The activated mast cell will now release its granules of histamine and heparin, thereby killing the parasite. In chronic infections with parasites, eosinophils play a role with their attempt at phagocytosis and releasing of their granules to kill the parasite. Unfortunately, IgE has a bad side: type 1 hypersensitivity reactions with allergies and anaphylactic shock. Once IgE binds to its cognate allergen, it will activate the mast cell with their IgE receptors. Just as you saw earlier with degranulation with the parasites, here you have degranulation, releasing of histamine and heparin, forming an allergic reaction in the atopic individual. Which cells are involved in type 1 immediate hypersensitivity reactions? Basophils. Type 1 hypersensitivity reactions involve IgE interacting with the allergen and then activating the mast cells with the release of histamine and heparin, forming the classical symptoms of allergies. Anaphylactic shock is the worst kind, and then you'll see later on chronically eosinophilia. Now for the light chains, kappa and lambda. The normal ratio is 2 to 1. If there's an abnormality in this ratio, 3 to 1 or 1 to 2, then this might point to a plasma cell neoplasm. Bence-Jones proteins are a result of a monoclonal gammopathy with the light chains found in the urine, and this is suggestive of multiple myeloma or Waldenstrom's macroglobulinemia. We'll look at the parts of the antibody molecular structure of the variable region, which is antigen specific. The variable region also is known as the antigen binding region, FAB. The antigen binding region binds to the epitope, which is the antigenic determinant that is recognized by the antibody. Antigen binding regions also binds to specific haptens, which are small molecules which are combined with a carrier molecule that can elicit the production of antibodies, but the hapten cannot elicit antibodies by itself. Idiotype is a shared characteristic between a group of immunoglobulins or T cell receptor molecules. They are part of the same family because they bind to the same epitope and they share the same variable region, or the VL and VH domains of the variable regions are identical. The constant region of the heavy chain is shown in blue. It includes the FC region or crystallizable region. Antibody domains has been presented with VH and VL for variable heavy and variable light domains. Also, CL for constant light and CH1, 2, and 3 for constant heavy 1, 2, and 3. I saw one question in the ASCP study guide that asked for the location of the hinge, which is between CH1 and CH2. Papain is an enzyme that cleaves the antibody into three parts: two FAB segments and also an FC, which is crystallizable segment. Our amazing antibody idiotypic diversity is obtained through the magic of recombination of the B cells in the germline theory, and more to be followed later in other videos of this fascinating topic. The following questions and answers are similar to the ones in our ACP study guide, but not exact, but the concepts are similar.